Good morning again. So, we are again coming to the same topic of diffusion, and let's define it together that it is a net movement of particles from an area of high concentration to an area of low concentration. Please remember one thing, that these particles over here could be of any state of matter. It could be solid, it could be liquid, it could be gas. Okay, so anything, any particle, it could be solid, liquid or gas, it always starts moving from a region of its higher concentration to the region of its lower concentration. ये फैलना शुरू करेगा, जहाँ पे ये ज़्यादा है वहाँ से दूसरी जगहों में. ये तो हम नॉर्मली भी देखते हैं कि फॉर एग्जाम्पल इन योर क्लासरूम at one corner there are 15 to 20 students sitting, so it will be very difficult to be there. So what will happen, you will start moving to those places, those corner or those places of the classroom where there are less number. So it's a universal kind of phenomenon. Second, it is due to the random movement. That means there is no precise direction where it should move. It can move in any place because these particles are having kinetic energy with them, so they will have energy to move in random direction. This is a passive process. It's a passive process. That means externally no energy is required to be given. It can use its own internal kinetic energy. No amount of energy we have to provide so that the particles can start doing diffusion. So diffusion is important to understand in bio also, because it explains the movement of many different particles. For example, the movement of oxygen and carbon dioxide in and outside of the cell. Let us try to understand it by taking the example of amoeba. So the gaseous exchange that take place in the amoeba, that's a unicellular organism, is through the process of diffusion. That means the oxygen gets inside the amoeba body and the carbon dioxide produced during respiration gets outside the amoeba through the cell membrane by the process of diffusion only. Now, as I have told you, diffusion can be for gas, for solid or for liquid. So what causes this oxygen keep on entering? It is because the amount of oxygen present inside this amoeba cell will be very less in concentration or percentage as compared to the oxygen in the same volume in the environment. So obviously oxygen will undergo diffusion and start moving from its higher concentration to lower concentration area. तो ऑक्सीजन अपने आप कहाँ एंटर करेगा? अमीबा के बॉडी के अंदर, अमीबा के सेल के अंदर. Now, during this oxygen entry, this oxygen will be used for what? For doing the respiration process. And what is given out in respiration process? Carbon dioxide. That means the cell has a lot of concentration or high concentration of carbon dioxide. When we compare the same volume of environment for the carbon dioxide portion, it is comparatively less. So again diffusion process will help the carbon dioxide to move from the cell of amoeba outside it. So why does CO2 move out all the time? Because concentration of CO2 is very high in the amoeba and it is very less in the atmosphere, so diffusion will be there. Similarly, why does oxygen keep on entering? Because concentration of oxygen in the outer environment atmosphere is high as compared to the oxygen concentration inside the amoeba. So gases move down their concentration gradient. Gradient का मतलब है कंसंट्रेशन कितना ज़्यादा और कितना कम है. हमेशा गैसेस कैसे मूव डाउन करती हैं? हाई टू लो कंसंट्रेशन जाती हैं. In later senior classes you will understand that for gases we replace the word concentration with pressure. So later on we will understand that pressure of oxygen is causing it to happen. So let us come to the next topic over here. Now, after this diffusion, we are in a position to understand that yes, this is a cell membrane. If you remember the lipid membrane है, ये lipid bilayer है, दिख रहा है. ये वही लिपिड के मॉलिक्यूल्स है, टेल के साथ प्रोटीन्स भी होंगे. तो कोई भी मॉलिक्यूल अगर अपने हायर कंसंट्रेशन के साथ सेल के बाहर है तो वो क्या करेगा? वो सेल के अंदर एंट्री करने की कोशिश करेगा. क्यों? वहाँ उसका कंसंट्रेशन कम है. फॉर एग्जाम्पल, इमेजिन दिस पार्ट, द राइट साइड इज द कंप्लीट सेल. left side is the environment. So these green molecules, it could be anything, it is having higher concentration over here and it is very lower concentrated over here. So what will happen? Diffusion will cause it to start moving from its high concentration to low concentration. Now, diffusion has a very special case, and the case or the property here is known as osmosis. Now, osmosis is a special type of diffusion. मतलब यहाँ भी any molecule will start moving from its higher concentration to लोअर कंसेंट्रेशन. ये तो सेम रहेगा, तो डिफरेंस किसका रहा? यहाँ पे क्या होगा, हम सिलेक्टिवली सॉल्वेंट पार्टिकल के मूवमेंट की बात करेंगे. दैट मीन्स हम यहाँ पे सॉल्यूट की बात नहीं करेंगे, हम किसकी बात करेंगे? सॉल्वेंट की, जैसे कि वाटर के मूवमेंट की. ठीक है. दूसरा व्हाट वी आर गोइंग टू अंडरस्टैंड दैट between the two concentration there should be present a semi permeable membrane. यहाँ एक semi permeable membrane होना ज़रूरी है, जैसे कि cell membrane. That means osmosis is a property in which any solvent molecule, that means water, moves from its region of higher concentration to the region of its lower concentration. Now try to understand these molecules over here. Can you see the left side of the through a semi permeable membrane? Left side का आपको बीकर दिख रहा है, लाइट ब्लू कलर वाटर को डिनोट करने के लिए दिया. Can you see this small molecule? This represent water. So if I count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 13 to 14 water molecules are present here. इस सेमी परमेबल मेम्ब्रेन के राइट साइड पे देखिए, यही वाले वाटर मॉलिक्यूल्स कितने हैं? 1, 2, 3, 4, 5, 6, 7, 8. दैट मीन्स वाटर यहाँ पे ज़्यादा है और वाटर यहाँ पे कम है. तो आपका क्वेश्चन होगा, मैम ये ब्लू वाले बड़े बबल्स क्या हैं? दीज आर द सॉल्यूट्स. सॉल्यूट जैसे कि हमने एक वाटर का सोल्यूशन बनाया, तो वाटर क्या कहलाया जाएगा? सॉल्वेंट कहलाया जाएगा. उसके अंदर जो हम शुगर मिला रहे हैं वो उसका सॉल्यूट है. तो ऑस्मोसिस में हम शुगर के मूवमेंट की बात नहीं करेंगे. वी विल ओनली एंड ओनली टॉक अबाउट द मूवमेंट ऑफ द वाटर मॉलिक्यूल. सो वाटर इज प्रेजेंट मोर ऑन दिस साइड, सो इट विल स्टार्ट क्रॉसिंग दिस सेमी परमेबल मेम्ब्रेन एंड मूव टूवर्ड्स दिस साइड. सो ऑस्मोसिस में इंपॉर्टेंट क्या है? इट इज अ स्पेशल टाइप ऑफ डिफ्यूजन वेयर solvent or liquid water molecule starts crossing and moving from its higher concentration to its lower concentration through a semi permeable membrane. तो water के इस movement को और समझने के लिए we will go for the next topic. So first of all let us try to understand that what is a solution. Remember, अगर हम कोई भी solution पढ़ते हैं, क्लास 9 की केमिस्ट्री के टॉपिक में भी आप पढ़ेंगे, solution has two main component. One is the solvent, the blue color part, that is for example water, and these particles or bubbles over here you are seeing, observing, क्या आप मेरे एरो को देख सकते हैं? These are the solute. अगर define करें तो solvent means a liquid in which substances, for example solutes, are dissolved to make a solution. ठीक है. solute क्या है? Solute is a substance that can be dissolved by a solvent to create a solution. That means solvent वो है जिसके अंदर हम dissolve करते हैं solute को, और solute क्या है? वो particles हैं जो solvent के अंदर dissolve होते हैं. solute का example क्या हो सकता है? For example sugar solution बनाया आपने, solvent क्या होगा? water होगा, and sugar क्या होगा उसमें? solute. आप salt solution बना रहे हैं, lemonade बना के पी रहे हैं, what will be your solvent? It will be water. And what will be your solute? It will be your salt, sugar, everything that you are adding. In a way we can say एक solution में solvent का amount थोड़ा ज़्यादा लिया जाता है और solute का amount comparatively कम लिया जाता है. So let us try to understand different kinds of solution. Now, this solution can be subdivided into three types of solution. One word is known as hypertonic solution. Second word is known as hypotonic solution. Third word is known as isotonic solution. अब इस हाइपर, हाइपो और आइसो को समझने के लिए पहले हमें टॉनिक का मतलब समझ आना चाहिए. टॉनिक मेडिसिन को भी कहा जाए. टॉनिक मीन्स स्ट्रेंथ. कितना परसेंटेज सॉल्यूट उसमें है या कितना परसेंटेज सॉल्वेंट है. So basically what we are doing, we are comparing a solution kept in a beaker with the solution present inside a cell. अब सेल के अंदर कौन सा सोल्यूशन है भाई? सेल के अंदर सोल्यूशन को साइटोप्लाज्म कह रहे हैं हम, सेल सैप को कह रहे हैं, जिसमें वाटर भी होंगे, obvious साइटोप्लाज्म वाटर भी होगा, and along with that it will have certain solute particles also. So we are comparing the solution present in the beaker and the solution present in the cell sap. कब कंपेयर कर रहे हैं? जब इस सेल को हम इस बीकर के अंदर डाल रहे हैं. So we are comparing this solvent present in the cell with this solvent present in the beaker. Are you understanding? Similarly, we are comparing the solute present in the cell with the solute present in the beaker. So explanation of this kind of solution we are going to do in the third video for today.